Good morning, and first thanks to the organizers for the invitation today. I work in a computer science lab in the northeast of France, but I work in close collaboration with biologists from INRA in Versailles, close to Paris. And I'm interested both in technology as a computer scientist, but also in biology. The objective of my work is twofold. Understand olfactory coding in insects by using technology and by means of computational modeling, basically, and apply this knowledge about the functioning of the biological olfaction to develop artificial devices like electronic noses. So in the past, we've developed, in collaboration with Amin Bermak from the University of Science and Technology of Hong Kong, biologically inspired electronic nose. And the device consisted of 16 sensors and 16 neurons, so 1 neuron per sensor. And the neurons transform the sensor output into a temporal code that is a train of spikes or action potentials. So a given neuron will fire a spike at a time given by a nonlinear function of the sensor output. And in that function, there is an encoding parameter for each neuron that needs to be trained in advance in order to encode particular odors. So here is, I'm going to show you a short demo in which you see the device in action. So the electronic nose is in this gas chamber and is sniffing the vapor of the odor released by this in this container here. And the electronic nose will encode the odor into a spike train, which is transmitted wireless to a mobile phone for odor learning and recognition. So you see that the spikes arrive at different time because the sensors do not have the same activation level. And this pattern of spikes is representative of the odor that we want to encode. And the good thing is that this pattern here, the spike train of 16 spikes, because we have 16 sensors, is invariant to changes in concentration. So for example, for hydrogen here, for 25 to 75 ppm, we see that when we increase the concentration, then the entire pattern is shifted to the left because the neurons are more activated by the sensors that I fired earlier. But the relative timings between the spikes here are preserved. And it works for hydrogen, but also for carbon monoxide and ethanol. And this pattern of spikes here changes if we change the odor. So if we use carbon monoxide instead of hydrogen, we have a different pattern. Here we have the neuron 5 and 1 at the end. For example, here we have 4 and 7. So it's easy to recognize the odor by just looking at the pattern of spikes that is encoded by this device. Okay, so here we have a device that is biologically inspired and is simple enough to run on mobile phones, for example, to detect alarms when dangerous gases are detected, like carbon monoxide. And this is one example of translating neurophysiological finding into artificial devices. But today I would like to talk about the opposite. Use technology to apply technology to neuroscience and try to understand olfactory coding by using technology and technological devices. So the question I'm interested in is the following. How are odors encoded in the insect brain to control the behavior of the insect? And to answer this question, you will see that I'm using a combination of different techniques from electrophysiology to computational modeling, so simulation using computers, and also robotic experiments. So here is an insect flying in a wind tunnel and trying to locate the source of an odor. So this insect is a moth. The source of the odor is a pheromone, a sexual pheromone. And you first see that the odor is not continuous cloud, but it consists of patches, intermittent patches here, so that even when the insect flies within the plume, it may lose contact with the odor. And so its behavior will be affected. Actually, there are two kinds of behavior that have been identified. When the odor is perceived, then the insect do a straight movement upwind, which is called a surge upwind. And when it loses contact with the odor, there is a more complicated behavior called casting. We doesn't know yet what is behind casting, but at least there are two behavior. First, just after losing contact with the odor, the insect perform a crosswind, a zigzag crosswind, like here, in order to relocate here, in order to relocate the contact with the source. If it doesn't succeed, then there are more looping performed by the insect. So the specific question I would like to answer is, how are these odor patches encoded in the insect brain to have this switch between surge and casting behaviors? And to do that, I work in collaboration, as I said before, with biologists from INRA, and we recorded from neurons in this insect, in the mouse brain. And by opening the head of the insect and inserting a microelectrode, we can record neurons from the antennal lobe. The antennal lobe is the first processing stage of olfactory information in insect, and it simulates the olfactory bulb in mammals. And it's made of many neuropils called glomeruli, as you see here. And in this mouse, there are 3 larger glomeruli in gray here in the figure, and they are dedicated to the 3 main compounds of the pheromone. So what we did, we stimulated the antenna of the moth using a blend of these 3 main compounds, which is the sexual pheromone for this moth, and we are recorded from neurons that belong to these 3 glomeruli. When we do that, what we observe is the following. So the x-axis here represents time. The y-axis is the number of tries, because we have repeated the stimulation. And a single row here is the activity of the neuron that is being recorded in a given trial. The gray rectangle doesn't work. The gray rectangle here represents the stimulus that has been sent for 200 milliseconds. And you see that we have first an excitatory response that we call the on response, because it characterizes the presence of the stimulus and the duration of this on response. Yeah, because it doesn't. Yeah, it's better. Okay, so the duration of the on here is equal to the duration of the stimulus. So in that case, 200 milliseconds. Then if we stop the stimulation, what we have is, first we have an inhibitory phase of 400 milliseconds. And then we have more tonic excitation, which we call it the off response, because this response is induced by the absence of the stimulus. And it can be very long, more than 10 seconds. And you see that if we repeat this stimulation, then we have the same triphasic response: excitation, inhibition, excitation. And it's even more impressive because when we zoom on the on response and we look at the spikes, individual spikes over the different trials, we see that they occur approximately at the same time. The jitter is a few milliseconds only. So we have a very precise and stereotypical response for this neuron. And then we tested if this neuron can follow pulses of stimulus, because a moth flying in a plume will encounter these intermittent stimuli. So here we have pulses of stimulus, and we can see that the neuron responds with the on and inhibition, on, inhibition, on, inhibition. So it can follow pulses that are realistic. Then if we pharmacology blocked this inhibitory phase using a drug called bicuculin, then this is what we get. The neuron is not capable of following the pulses anymore. So this shows that we need the inhibitory phase to be able to encode the odor information that is intermittent, as encountered by a mouse flying in a realistic situation. Okay, so this is the effect of bicuculin in the neuron. So at the beginning, time t equal to 0, we clearly see the on and inhibitory phases in the response. But as the drug diffuses in the tissue here, we see that the inhibition vanishes. And after a few minutes, then what we have is a pure tonic response here. And this process is completely reversible because after washing, then the neuron has still recovered its capacity to follow the pulses. So the bicuculin is known for mammals to block small conductance calcium-dependent potassium channel. So that's an ionic channel which is internal to the neuron. And the hypothesis we made is that this shaping of the response is induced by internal properties of the neuron, not by the network and the interaction with other neurons. It's more due to intrinsic channels. And it's quite complicated to prove that in biology because there are no specific blocker of this SK channel for insects yet. So we perform a model, a Hodgkin-Huxley model of this neuron that incorporate an SK channel, and we try to reproduce the biological data using this model. So here is the model, and then these are the comparison between the model and the experiments, and they fit quite well. So we have, with the model, we still have this inhibitory phase of 400 milliseconds, and we have the duration of the on, which is equal to the duration of the stimulus. If we block this SK channel in the model, then we lose the inhibitory phase as the biological experiments with the bicuculin. So this was our first hypothesis, whether the SK can be involved in shaping the response of the neuron. The second hypothesis is that can SK, this channel, control the behavior of the mouse? And to test that, we use a robot and our neuron model as a motor, as a common neuron. So the activity of the neuron model will trigger distinct surge and casting behavior. But to do that, we need some sensors, pheromone sensors, and we use the mouse for that. So we took a mouse, put it on the robot, and use its antenna. The antenna of the mouse is the nose for us. And we use two electrodes, one electrode on the tip of the antenna, the other electrode, which is the reference on the base, on the neck, more precisely, that we have an electrical signal, which is called the EAG for electroantennogram, and which reflect the activation of the nose of the mouse, which is the antenna. And this electrical signal is sent as input to our neuron model, which runs in real time. So in red, you have this antennal output, and in green, you have the neuron model output that has this as input. And we detect on this neural output here the on and off activity to trigger surge and casting. And we perform experiment to see whether we can find the source using this model, and we will do experiment by blocking the SK and not blocking and having SK intact. Okay, so the difficult part was to specify the casting of the behavior, because the surge is pretty easy. It's just a straight movement upwind. But the casting, as I said, is more fuzzy. So what we did is the following. So here is the neural response. We have first excitation, which is the on, which signals the presence of the pheromone, trigger the surge. Okay? Then we have the inhibitory phase, the on and baseline. Baseline is the baseline activity of the neuron. So first we consider one-step casting. So the on will be considered similar to the baseline. And both off and baseline will signal the absence of the pheromone and will trigger some exploratory behavior. And we choose some looping behavior. Why looping? Because the source cannot be missed using this kind of strategy. And theoretically, we can show that the maximum search distance to find the source will be 22D, where D is the shortest distance from the source at the beginning of the search. So we have some ground, and here is some video showing the cyborg in action. So first time we have SK channel intact. You see the on-off neuron model that has as input this electroantennogram. And you see that, so here there are detections, and you see when there are detections, there is the on and inhibitory response on the neuron. The source is here. The robot starts at 2 meters. The wind flows in that direction. And there are more detections when the robot approaches the source. We compare with and without the source. I mean, it's quite robust. Here is the without SK in the channel, without the SK channel in the neuron. And so the neuron doesn't have this on and inhibitory phase anymore, so we don't detect the surge, and it does casting all the time because the baseline activity. So even when we have some detection by looking at the sensor signal, which is the output of the antenna, we don't have this on response on the neuron model. So here is the comparison between what we have in the cyborg and mouse experiments, which have been done by another team. And so here are the cyborg experiments. We see that this is the histogram of the track angle. I don't want to go into detail, but there are some similarities between real mouse trajectory and what we get in robotic experiments. This is in case of SK channel intact. This is when the SK channel is blocked. Then we see that the track angle histogram is more flat, which is also similar to what we get when moths are flying, when moths are treated with bicocline and they're flying in the wind tunnel. It affects their behavior. And then with SK channel intact, we found the source in more than 90% of the case, where the SK blocked, we found the source by chance only in less than 10%. And the search distance when we found the source with SK intact is 4 meters. The average where is more than 15 meters in case of SK is blocked. Okay, so in this second part, we consider a two-step casting strategy by separating the off from the baseline. So the baseline will do as before. It will signal the absence of the stimulus and will do looping behavior. The off response of the neuron will signal the loss of the pheromone. It means that you have seen the pheromone or the odor before, but you lose it. You lost it, sorry. So, it's because it's not very far. Maybe a better strategy will be to zigzag crosswind in order to relocate the plume sensor line. So this off activity in the neuron will trigger another casting, which is a zigzag crosswind. So we have two casting now. And still, if we analyze theoretically, then we show that in that case, the maximum search distance will be 9D, which is smaller than 22D that we have before with the single casting strategy. If we do the experiment with the two-step casting, then this is what we have. So here there is a search, then casting the zigzagging crosswind, then it doesn't find, and there is this looping. And by repeating the experiments, we show that this two-step casting outperforms one-step casting in terms of search distance. So the search distance is smaller, like 3 meters versus 4 meters. So to conclude, so in the first part, I've shown that I've used some understanding of the biological coding to develop biologically inspired electronic noses. So I've applied, I've transferred knowledge about biology into artificial devices. In the second part, I've used technology, and I've applied technology to understand olfactory coding. The technology was computational modeling of a single neuron using this SK channel to reproduce the electrical activity of the neuron, and the use of robotic experiments to test behavioral hypotheses. And these are really two sides of the same coin. Using technology to understand biology, and using biology and get inspiration from biology to develop artificial devices. And more specifically, our modeling results suggest that a single neuronal property, which is this SK channel, may account for the triphasic firing pattern and the complex behavior of the insect. But it's not a proof, and it needs experimental work in biology in order to prove that. And the hope is that this, in Drosophila, a single gene has been recently identified for coding for this SK channel. And this gene was expressed in the same olfactory neuron that we recorded in the mouse. So it will be interesting to knock out this channel, this gene in Drosophila, and observe whether it has an effect on the electrical activity of the neuron or on the behavior of the Drosophila. Okay, so I stop, and I thank you for your attention.
Thank you.